Hi friends, in this video let us learn about recombinant DNA technology. In 1963, an enzyme was found to restrict the growth of bacteriophage, and that enzyme was later known as restriction endonuclease. Okay, restriction endonuclease. Okay, so we're going to learn about this particular enzyme. This is also known as molecular scissors. Okay, now we have about 900 such enzymes. RE means restriction endonuclease. We have 900 such enzymes. Okay, and these enzymes have been derived from some 230 strains of bacteria. 230 strains of bacteria. So our topic is recombinant DNA technology. So in 1963, an enzyme was found. The name of that enzyme was restriction endonuclease. It was able to restrict the growth of bacteriophage. That is how it was first isolated. Later on, they named it as restriction endonuclease enzyme. It is popularly known as molecular scissors. Now we have around 900 such enzymes taken from 230 strains of bacteria. Okay. So these enzymes, what they will do? They actually cut DNA at specific sites. They cut DNA at specific sites, right? Actually, the enzymes present in nuclease are called nucleases. Okay, we have exonuclease and endonuclease. Okay, this exonuclease will cut DNA at the ends. Okay, supposing this is the enzyme, these enzymes will cut DNA at the ends like this. Okay, whereas this endonuclease will cut within the DNA. Okay, endonuclease, as the name tells, it will cut DNA at the within the DNA. Okay, in between the DNA it will cut. So there are two kinds. One known as exonuclease removes DNA at the ends. Endonuclease cuts DNA within the DNA at specific ends. Okay. Let us see how these enzymes are being named. Okay, how restriction endonuclease are being named. Okay, example in E. coli. How it is being coined in E. cor 1. We'll see that how it's being coined. Okay, this is an restriction endonuclease enzyme E. cor 1. Okay, so this letter E stands for Escherichia. It is the genus of an bacteria, the genus name of a bacteria. Okay, and the CO, it came from coli. Okay, all know that the species name of Escherichia is coli. So E. coli. So this CO stands for coli. R denotes the strain of bacteria. Okay, whether it's a rough strain or smooth strain. Okay, it denotes the strain of bacteria. And this is not I, that is Roman letter 1. Okay, this is Roman letter. It is the order in which the enzyme has been isolated. The order in which the enzyme has been isolated. That is how the names are being coined with a specific reason. Now I have given an example how restriction endonuclease, that enzyme ECOR1 is being named. E stands for Escherichia, the genus of the bacteria. CO stands for coli, the species name, 2 letters of the species. R stands for the strain of bacteria, whether it's rough or smooth. And this Roman letter 1 stands for the order in which it is being isolated. Okay. Now we'll see the functions of whenever I write RE, it is restriction endonuclease. Okay. Restriction endonuclease. Okay. First of all, this enzyme will inspect the entire DNA strand. Okay. First thing is inspection. Okay. Second, it finds. Okay. Specific recognition site. It finds a specific recognition site. Then it binds. It binds to that DNA. Okay. Then 4, it cuts. First, the restriction endonuclease enzyme, it inspects the entire chain of DNA. It inspects. Then it finds a specific recognition site. And third, it binds to the DNA. And fourth, it cuts the DNA strands. And something about the specific recognition site. This site will be of palindromic in nature. Okay, palindromic, a word which can be read forward and backward. So that specific sequence, recognition sequence, will be of palindromic in nature. For example, if this is the 5' end, if this is GAATTC, this is a sequence. The palindromic will be like this. Okay, CTTAAG. Okay, I'm talking about the palindromic sequence. Okay, palindromic means a word that can be read forward and backward. It should be same. Here, this restriction endonuclease will select a specific sequence based on palindromic nature. Here is an example. This is a DNA strand starting from 5' to 3' GAATTC. You all know that guanine, adenine, thymine, all these things. Okay. The same thing in reverse order, 3' to 5'. Okay. Guanine, cytosine, adenine, thymine. You know this. So this is the sequence. So this kind of palindromic sequence will be selected, recognized by the restriction endonuclease. Now a schematic representation how rDNA is being formed. Okay. So this is the foreign DNA. Okay. This is vector DNA. Okay. Vector DNA will be plasmid. Okay. So they are being cut by restriction endonuclease. Okay. They are cut. And they are attached by means of ligase. Okay. DNA ligase which attach them. Okay. Then we have the newly formed recombinant DNA. Okay. And that will be transformed into the host cell. For example, E. coli, where it gets multiplied. So this is the schematic representation of recombinant DNA technology. Recombinant DNA technology. Foreign DNA, vector DNA, that is plasmid. Both the DNAs are cut by restriction endonuclease and they are attached by means of DNA ligase. A newly formed recombinant DNA is there, which is being transformed into the host cell. Okay. That is a schematic representation of formation of rDNA. But how to separate that DNA fragments? How to separate and isolate DNA fragment? So that is not an easy thing. So let us see how it is being separated and isolated. Okay. Separation and isolation of fragments. So it needs electric field, and that is known as gel electrophoresis. Okay, DNA are negatively charged. So they have to move towards anode. They have to move towards anode. So we need a device called gel electrophoresis on which the medium is being applied. The medium or matrix is agarose gel. It is a natural polymer obtained from seaweed. So what is agarose gel can be asked for a 1 mark. It is a natural polymer obtained from seaweed. Okay. So on this agarose gel, the DNA tends to move towards anode. DNA are negatively charged particles. Okay. Then so this DNA fragments will keep moving. The smaller the size, the farther it moves. Okay. So smaller DNA fragments will move and reach the destination earlier. And for this, to visualize, we have to add a stain called ethidium bromide. Okay, and that is followed by UV exposure. So to visualize the DNA fragments, we need a stain called ethidium bromide, which is exposed to UV rays. Then you can see the DNA fragments, especially in bright orange color bands. Okay, in such a way you can see the DNA fragments. Actually, you can visualize orange color DNA fragments as bands. Okay. So this is a technique. And later, the DNA fragments are extracted from the agarose gel. That particular process is called elution. The separation of DNA fragments from agarose gel is called elution. And that is how the DNA fragments are being isolated and separated. So it involves a technique. The technique name is electrophoresis. It's a device, okay, where electric field is applied. It needs a medium or matrix, and that is called the agarose gel. That agarose gel is taken from a natural polymer, seaweed, okay, and that particular medium has a sieving effect through which the DNA passes through. The DNA passes through. Smaller the size, farther it moves. So it moves towards the anode as DNA are negatively charged, right? So through the sieving effect, it moves, okay, and we need a stain called ethidium bromide to visualize DNA fragments. And that ethidium bromide, after applying that, we need to expose it to UV rays. Then you can see orange color DNA fragments. Okay. Later on, from that agarose gel, it can be extracted. That process is called elution, and that is how the DNA fragments are being separated and isolated. So with this, we come to end of our session. In next video, let me explain to you about cloning factors. Thank you. Hope you like this particular video. Please share and subscribe. Thank you.